Yes, it is.
Okay, so hello everyone. My name is Amarpreet Brar, and my research project focuses on utilization of nanoparticle-enabled combination therapy to overcome antimicrobial resistance from in pathogenic bacteria isolated from swine farms. So we all know that antibiotics have been developed for the intention of usage in humans. However, today about 70% of antibiotics are actually used in animals for various reasons, such as infection treatment, prevention of infection, as well as growth stimulus. However, due to overuse of antimicrobials in food animals, this can lead to emergence of antimicrobial resistant pathogens, which can then be transferred to humans through the food chain by consuming contaminated food or unsafe handling of the food. And these infections can now require high dosage of antimicrobials or even alternative therapeutics. Antimicrobial resistance causes a huge burden on the economy, the food industry, as well as the healthcare. It's estimated that by 2050, about 10 million deaths per year will be caused due to antimicrobial resistance, which is same as the combined deaths due to cancer and diabetes. Therefore, it is very evident that there's a need to introduce alternative therapeutics so that the amount of antimicrobials that are being used can be lowered down, and therefore we can slow down the process of emergence of antimicrobial resistant pathogens. Such alternative therapeutic is nanoparticle enabled combination therapy. Nanoparticles are very small in size, therefore they can penetrate deep tissue layers and can deliver drugs intracellularly. They also provide increased stability to the encapsulated drugs or the components due to various harsh environments of the gastrointestinal tract or the enzymes, etc. Nanoparticles can also be functionalized on their surface with various ligands to target specific cells or tissues inside the body. And also, more than one component can be delivered simultaneously using nanoparticles. And controlled drug release at the site of infection is another advantage offered by nanoparticle-enabled combination therapy. Moving on to my research and my results. For this purpose, 7 bacterial isolates were isolated from swine farms across Quebec, which consisted of Staphylococcus aureus, Staphylococcus haegus, Salmonella enterica cerevisiae, Typhimurium, and Cholera suis. These are bacteria that cause various infections in swine and as well as also cause various foodborne infections in humans as well. Therefore, there's a need to understand these bacteria better. And the first step is to understand the antimicrobial susceptibility of these bacteria to various commonly used antimicrobials. And this was done using Kirby-Broad disk diffusion assay. And it was found out that all the tested bacterial isolates were multi-drug resistant, and Salmonella typhimurium was resistant to almost all the tested antimicrobials. It was also seen that a high frequency of antimicrobial resistance was seen against commonly used antibiotics such as ampicillin, lincomycin, spectinomycin, tetracycline, ticarcillin, etc. Now, the next step would be to understand the resistant mechanisms of these bacteria better. The first resistant mechanism that was tested was efflux pumps. Efflux pumps are proteins on the surface of the bacterial membrane which help in extrusion of toxic components, herein antibiotics. Etherium bromide was used to test the efflux pump activity of these bacteria, and it was found out that except S. haegus M48, all the bacteria possess high efflux pump activity as well. The next resistant mechanism that was tested was beta-lactamase enzyme activity. Beta-lactamase enzyme is responsible for hydrolysis of beta-lactam drugs such as penicillin or ampicillin. For this, nitrocephin hydrolysis was used to measure the beta-lactamase enzyme activity, and it was found out that except S. haegus M48, all those tested bacterial isolates possessed high beta-lactamase activity as well. So now that the resistant mechanisms were understood, the next step is to determine suitable combinations of antibiotics and adjuvants that can circumvent these resistant mechanisms and inhibit the growth of these resistant bacteria. For this, checkerboard assay was used in which compound A, which could be the antibiotic, was diluted column-wise, and compound B, which could be the adjuvant, was diluted row-wise, and the minimum inhibitory concentration of the combination was determined, which then was used to categorize these combinations into synergistic, additive, indifferent, or antagonistic effect. For the project, ampicillin, chlorhexidine, tetracycline, trimethoprim were tested in combination with various adjuvants against Salmonella enterica cerevisiae typhimurium and SRES M12. And it was found out that tazobactam and ampicillin had synergistic effect against both of these bacteria. And this is because tazobactam is a beta-lactamase enzyme inhibitor and ampicillin is a beta-lactam drug. Synergistic effect was also seen in combination of cinnamon essential oil and ampicillin and oregano essential oil and chlorhexidine against Staph aureus M12. And this is because essential oils in general perforate the bacterial membrane, which allow easy passage of the drugs into the bacterial cells, thus killing them. Now that the suitable combinations are determined, however, these combinations cannot withstand the harsh environment of the gut or various enzymes, etc. The next step was to determine suitable nanoparticle-enabled drug delivery systems suitable for these combinations. First nanoparticle that was tested was liposomes for the co-encapsulation of hydrophilic drug tetracycline and hydrophobic cinnamon essential oil. The outer part of the liposomes, it consists of the hydrophobic part, and the inner core consists of the hydrophilic part. This has various advantages, such as the ability to co-encapsulate the hydrophobic and the hydrophilic drug, and is very economical and easily available. However, there are some drawbacks, such as the hydrophilic drug is encapsulated in very low quantity because of the small hydrophilic core, and also the cinnamon essential oil, instead of being encapsulated into liposomes, can sometimes form emulsification, thus hinder the formation of liposomes itself. Another nanoparticle drug delivery system that was tested was chitosan. It has various advantages, such as a positive surface charge, which makes it mucoadhesive. It's also very economical and easily available. Also, it has high encapsulation efficiency for both hydrophilic and hydrophobic drugs. And most importantly, it's biodegradable, biocompatible, and non-toxic. Nanoparticle drug delivery system that was tested was silica nanoparticle. Silica nanoparticle also has various advantages, such as high number of synonol groups on the surface, which allow for easy functionalization of this nanoparticle. It also has a large number of pores and surface area, which allows for large encapsulation of drugs. And similar to chitosan nanoparticles, it's also non-toxic, biocompatible, and biodegradable. Therefore, based on this information, chitosan and silica were selected for further experimentation. For the project, tetracycline was encapsulated into chitosan nanoparticle, and chlorpromazine was encapsulated into silica nanoparticle. Chlorpromazine is an efflux pump inhibitor, and tetracycline is inhibited by efflux pumps. So when these nanoparticles were combined in 1-to-1 ratio, this combination was tested for its antibacterial efficacy against the most resistant tested bacteria, which is Salmonella enterica cerevisiae typhimurium. And it was found out that tetracycline in its pristine form had MIC of 250 microgram per ml, and chlorpromazine in pristine form had MIC of 125 microgram per ml. But when these components were encapsulated into nanoparticles and then combined in 1-to-1 ratio, the MIC of the combination was 250 microgram per ml. Now, the encapsulation efficiency of chlorpromazine was found out to be 44.5%, and of tetracycline was found out to be 23.9%, which means that this 250 microgram per ml of the combination consists of 125 microgram per ml of the chitosan nanoparticle containing tetracycline and 125 microgram per ml of the silica nanoparticle containing chlorpromazine. Now, if you count the encapsulation efficiency, it means that only 20.32 microgram per ml of the tetracycline and 55.62 microgram per ml of the chlorpromazine, when encapsulated into the nanoparticles and then combined together, had the same effect as 250 microgram per ml of the pristine tetracycline and 125 microgram per ml of the pristine chlorpromazine. thus showing the enhanced antibacterial effect due to this combination and encapsulation into the nanoparticle. For future work, the effect of these nanotherapeutic will be tested on resistant mechanisms of the bacteria. Also, the antibacterial efficacy will be tested in vivo in C. elegans model, and this will help us to understand the interaction with the host as well as the bacteria itself. In conclusion, the tested pathogens were multi-drug resistant with various resistant mechanisms such as efflux pumps and beta-lactamase enzyme activity. And we also found that certain combinations of antibiotics and adjuvants were successful in overcoming the resistant mechanisms and therefore inhibiting the growth of these bacteria. And lastly, nanoparticles provide increased stability to the encapsulated drugs and can make them more effective against the pathogens.